So the session is recording. So majority of the students may prefer to watch the recording. I think that may be the reason
Yeah.
they didn't turn up today. Um, so we'll start with Sumit sir. Uh,
Okay.
I'm welcoming Sumit sir to CIAS Research Center. So a short yeah before handing over to sir, let me introduce. Uh, So, so I'm getting few messages. Okay. Dr. Sumit Mittal, he received his PhD in computational chemistry from Max Planck Institute, Germany. So and he did his BSMS program in chemistry from ISER Mohali, India. And his major research domain is applications of multi-scale molecular modeling and large-scale computer simulations to gain insight into structural properties of biological systems. So with this short intro, I'm welcoming Dr. Sumit to the forum. And over to you, sir.
Yeah, hi. Thank you. So I'll just directly jump to the project. So the project is about modeling the aggregation pathway of transthyretin. It's a protein which has a lot of functions in the body, but it's mainly interesting because it's associated with amyloidosis. So basically, what happens in this with this protein is that in usual normal conditions, it's present in this tetrameric form. So it's basically like a dimer of dimer, and it is pretty stable. But what was observed that under certain pH conditions and also with certain kind of mutations, this tetramer can be has been found to link with some different kind of diseases. And this is one potential mechanism that first the tetramer is dissociated into dimers. So basically, two of the dimers are dissociated, which then leads to the formation of monomers, folded monomers. They are still pretty stable, but these monomers from there on, they can undergo misfolding. So rather than go back to the original state following like the reverse side of the equilibrium, they are misfolded. And once these are misfolded, then it has been observed that they form some kind of oligomers. And these oligomeric forms are the ones which are considered to be pathogenic or sort of responsible for these different kind of diseases. So the exact nature or the exact form of the oligomers which are responsible for this, it is still not clear, like whether it is the tetrameric form or the octamer or dodecamer or so. And then in the patients, basically what is observed is that the fibril form of the protein is are observed. So basically these oligomers, they undergo the self-assembly process. They are organized, and rather than being organized in this tetrameric form, they adopt this fibril structure. And this fibril is the one that is found in the patients who suffer from different kind of diseases. So different kind of mutations, they lead to different kind of diseases. They have different impact on the body, and it's expected that the mechanism or the exact form of the oligomer which is responsible or which is found or which is sort of pathogenic for different mutations may also be different for different mutations. So this is what I propose to study, that we can start with the wild type or the native form of the monomer. And this monomer, we first study that how it is, whether it is stable, how it unfolds, and then this is the main step, that we want to see that what is the stability or what is the relative stability of different oligomers, and if we take like 12 monomers and we allow the self-assembly to happen, then which oligomeric form will be formed. And later on, obviously this can be like an extension. We can see that how these oligomers can lead to the formation of fibrils. But this is slightly complicated. So normal MD simulations, short MD simulations will not be able to account for this self-assembly. So there are different approaches that we can undertake. First is that either we can run like long MD simulations, multiple copies of the long MD simulations, microsecond or so, and then we can see that what are the oligomer forms that are stable within that time frame. Second approach is that we can take some enhanced sampling MD simulations, so Gaussian accelerated MD simulations or so, or Hamiltonian replications and these simulations. There are several other methods which are suitable for this process. Like so, we can uh, in relatively shorter time simulation, we can get much more information about the conformation landscape and the how the oligomers are formed as well. And for this, the software that will be used, we can either go with GROMACS or NAMD, so whichever the candidate is comfortable with. Then, and for the analysis, we will rely on MD analysis or different GROMACS or VMD utilities. So this is the idea. If you have any questions, then please feel free. So any questions or anything that's not clear or
So as we have only one participant here, Prabhupada,
Mhm.
if you have
Yeah.
any question, you can ask. Uh, Otherwise, we'll move to the next presentation. I think Prabhupada already contacted both of you, I guess. Uh,
Yeah, I got an email, but by then I didn't know like what's the how to respond. Or I was not familiar with the process, so
okay, okay, okay. It's
I'm
okay. Yeah,
happy,
yeah.
yeah, I'm happy to take any questions. So I think this is what you asked in the email also, like what is
Definitely,
the
definitely.
nature of
Yes,
the
yes, yes.
project. So I think you have some idea now.
Yeah, even Prabhupada also contacted us. So he also would like to attend this meeting to understand how this. I mean, he also wants to see this thing. What is the presentation? So maybe after the presentation, he can decide the things. Um, no, actually, Prabhupada, you are not audible. Or-
You can also type your question in the chat box. I
Ha ha
can
ha.
see.
Yes,
Yeah,
yes,
yeah.
yes, yes. I think he has some problem. Yeah. Yeah, the actually that question, I think I can answer that question, I guess. See, especially these two projects, we are trying to access one external HPC service. So maybe if you have if the calculation is bigger, or if uh, I mean, the fact is HPC, we are going to take that access of HPC, and you can use it remotely, that HPC. So most of these two projects calculations, we do it in HPC server. But yeah, that doesn't mean that all the
That
calculations you have to do in HPC. It's good if you install a few of the softwares, or maybe all the softwares in your system, and all the analysis you may have to do it in your system, and maybe a few of basic calculation, even the especially in the learning process, you have to use your computer. Yeah.
I think, but also 24 GPU. Depends on the GPU model, but I think it should be, yeah, as you're saying that for the timing, I think that will be sufficient. Uh,
Yeah. But yeah, as I said,
yeah,
the
this is
HPC has some limitation. The HPC that, not limitation, but it is kind of little um, budgetary thing. We may have to spend a little more money for that. So the basic. Uh,
very good GPU. It seems
Yeah, this RTX 4090 is the latest in the MD simulation. So
one of the latest release
yeah,
here.
one is more than
Yeah,
sufficient.
interesting.
Yeah.
Yeah. So, yeah, then in that case, shall we move to Bibop's presentation?
Yeah, fine.
Yeah. Uh, Dr. Bibab, it's very happy to introduce my colleague, Dr. Bibab. Uh, he is currently working as assistant professor in Department of Chemistry at VIT University. He received his PhD from Ruhr University, Potsdam, Germany, if I pronounce it correctly, and his masters from IIT Bombay. Um, so with this short intro, he also a computational chemist at VIT University. So with this short intro, I'm welcoming Dr. Vibhav to the forum. So over to you.
Yeah, thank you, sir. Thank you, Savan, sir. So, yeah, am I audible and is my screen visible, sir?
Yes, sir. Perfect. You are
Yeah,
good to go.
okay. So, as I have all proposed, I think two projects. So, I'll just start with one and then move on to the next one. So, thank you for this introduction, sir. So, the first project that I have proposed, that the title is approximately the understanding of the interaction mechanisms of antimicrobial peptides and bacterial membrane. So the idea is why do we need to study this kind of things? Because antibiotic resistance is one of the conditions that is a growing concern in the entire world. So because of the massive uses of antibiotics for multiple diseases, the evolutionary mechanisms of bacteria actually have made them resistant toward a lot of antibiotic medication. So that is the reason nowadays people are looking for different new molecules which are other than antibiotic drug molecules that have been used for so long. So the reason for having antibiotic resistance, for example, overprescribing of antibiotics, patients not completing the entire course of their antibiotics, unnecessary antibiotics used in agriculture, poor infection control, and so on and so forth. So a very real example or one of the most concerned example could be the multi-drug resistant tuberculosis, which is what is killing a lot of people these days. And a lot of research is going on to find out new medications for those kind of things. So antimicrobial peptides, the one thing that we are going to be using. So antimicrobial peptides are also referred to as host defense peptides that are part of innate immune system. And these are short peptides, 10 to 90 residues long, with a wide range of inhibitory effects against bacteria, fungi, etc. So these are the new generation antimicrobial molecules that has been shown to disrupt quite successfully bacterial cell membrane. They have also shown inhibitory effects against various bacterial enzymes, for example, angiotensin converting coenzymes and all, which are responsible for high blood pressures and all. So antimicrobial peptides have been successfully used and employed, and it is actually a constantly developing field for many applications. For example, wound healing applications, skin infection medication, agricultural application, etc. So these are some of the structures of antimicrobial peptides. They can be typically there. They have quite a lot of alpha helical and beta sheet content. A good number of well characterized secondary structures present in these peptides. So they can be short, like the first, for example, the short antimicrobial peptides. They can also be longer, like 40, 90 residues long. These the bottom two are the longer antimicrobial peptides chain. So what we are planning to do actually, so many animal trials and all these things are happening all over the world. People are coming up with many different antimicrobial peptide medications to study. So many of the studies have already been done, like how does antimicrobial peptide actually work against bacterial membrane and all. So, but some of the mechanisms are still not understood, and how can we develop a workflow to use molecular dynamics or computer simulation to study these particular phenomena. That is what this project is all about. So we'll use classical MD simulations to understand the intricate molecular interaction details between antimicrobial peptides and membranes. The structures of these bacterial inner and outer membranes can be modeled using MD simulations. They're actually very complicated structures, but some simplified model structures are available. And antimicrobial peptide structures like these can be built if their sequence of amino acid sequence are known using softwares like PyMol, VMD, and all. So this is roughly the workflow of the project that I'm proposing. So firstly, the candidate will undertake the project. They'll first have to understand working in Linux and installation of softwares like Promax, VMD, and other softwares in Linux environment, because Promax only works in Linux-based operating system. So the candidate who will be undertaking this project, they'll have to learn how to work in Linux-based operating systems and how to use Promax. So a little bit of command prompt-based analysis or how to type commands in Linux operating system, they'll have to learn. Definitely, I can clearly help in those things. Secondly, we'll have to identify a model bacterial membrane lipid compositions from available literature. There are plenty of literature data available that says what are the composition of different types of bacterial membrane. So we can try those different bacterial composition, membrane composition. Identification of antimicrobial peptide sequence from AMP database. For example, there is a very good database called DAMP. So where you can actually choose a particular function of an antimicrobial peptide, and it's an AI-based database. So it will give you the sequence, possible sequence, and their antimicrobial activity against some particular bacteria and all. And it will give you the it will give you the amino acid sequence. It will not give you the 3D structure. You can take the amino acid sequence and you can build the three-dimensional structure using some bioinformatics tools that we'll be using. And we'll actually, after that, we'll learn to use something called Martini model. So because these bacterial membrane simulations, they have a lot of molecules. So doing them using all atomistic MD simulation could be very tricky, especially if you don't have a very large high performance computing server or something. So what we will use, and this is a very already verified approach for this type of simulations, is Martini force field. It's a coarse grained approach where it's a 4:1 approach. For example, 4 atoms will be considered as one single bead or one single particle. So that reduces your computational costs significantly. So we'll learn how to use this Martini force field or Martini modeling, and we'll use that to model the membrane as well as the antimicrobial peptide. There are a lot of technical it is available, involved in that. We'll have to figure out all those things. And finally, once we have figured out the models, Martini models for the peptide and the membrane, we'll set up MD simulations of the membrane and the peptide using Martini force field. And we'll at the end analyze various membrane stability parameters to understand if the antimicrobial peptides are going to affect the stability of the bacterial membrane. So this is roughly what this particular project entails. So if someone has a basic idea of Linux operating system and all, it's very good. Otherwise, it's not a problem. We can first few days we can study the Linux environments and Gromax, and then these things will set up slowly as we move forward. So this is the first part of the project. Second project that I have proposed is the in silico screening of inhibitors for tyrosine kinase enzymes using MD simulations and free energy calculations. So this is a standard project idea for a computer-aided drug design type of thing. So I've chosen a particular enzyme, tyrosine kinase, because these are some of the most widely studied enzymes for a long time, because these are these enzymes are overexpressed in malignant cells and cancer-causing cells. So tyrosine kinases are actually a particular type of kinase enzymes. So they belong to a family of enzymes that transfer high-energy phosphate groups from ATP to substrate proteins. Tyrosine kinases actually do the phosphorylation in tyrosine residues of the substrate proteins. For example, on the left-hand side here, you have a tyrosine residue. After phosphorylation, its phosphate group is transferred to the tyrosine. So this is done by protein tyrosine kinases. So that is the reason the names are tyrosine kinases. Other names, for example, there could be serine kinases that actually phosphorylates the serine residue of substrate protein and so on and so forth. These enzymes are very, very important because these have been found in many malignant cell overexpression of kinases. So these things are found in cancer-causing cells. So this regulation of these enzyme activities have been identified as potential cancer-causing factors. So inhibitor search or some molecules, if we can search that can inhibit the action of or overexpression of these type of enzymes, then that can be treated as a potential inhibitory factor against cancers. And as I said, these have been done for a long, long time. I mean, it's not like something we're doing very new. People have been studying tyrosine kinases or different types of kinase inhibitor they have been designing for a long time. But so we have chosen one very interesting among these tyrosine kinases that is called Bruton's tyrosine kinase or BTK. It's a non-receptor intracellular signaling kinase. So there's a well-studied one for importance in its importance in the growth and malignancy of B cell. So it's an immune response cell, a white blood cell present in our blood. So these particular enzyme has a very important effect because it's involved in the intracellular signaling of B cells. So recent biomedical studies have also hinted towards the possible role of this kinase in the neurodegenerative diseases such as Alzheimer's disease. So there are already as I said, that tyrosine kinases have been studied for a long, long time. So there are some already approved FDA approved medication. FDA means Food and Drug Administration, US. So there have been a library of compounds, small molecules I'm saying, that have already been identified as BTK inhibitors. However, the impact of these inhibitors in various other pathological conditions, for example, lupus, autoimmune disease, neurodegenerative diseases, are not extensively studied. So this is the workflow that we are going to use. So there is an in silico process of screening these inhibitors. So we'll start with identifying some of those databases where FDA approved drug molecules are listed, or we can start with some online databases where many random drug molecules are there. For example, a database with 20,000 molecules or so. And from that, we can perform something called the molecular docking. So this molecular docking and another analysis called ADMET analysis, that is adsorption, desorption, excretion, toxicology study. So from that, we can actually get an idea of what are the possible molecules that can be used as a potential inhibitor drug for this particular inhibiting the actions of this particular enzyme. And so you see that after every level of screening, the number of possible target molecules are getting decreased. Okay, so that is the idea, that we'll start with a large number of possible uh, drug molecules, but we'll reduce it by using different methods. So at the end, our idea is to find using molecular dynamics, free energy calculations, and molecular docking to reduce the total number of hits to something around 10 to 40 or 50 like that. Then only this will be a physically meaningful approach. Okay, so this is something we are planning to do. So at first, we'll search for the different protein structural databases to identify a known three-dimensional structure of this particular enzyme and I understand its structural features and run unbiased classical all atomistic empty simulation. This time no Martini postural. Normal classical all atomistic empty simulations will run of this enzyme in water to understand its conformational dynamics and to obtain the minimum energy conformation of the protein. Then we'll use one of those library compounds. We'll have to understand how to look for those molecular libraries, how to download that many molecules, what are the file types and all those things. And we'll have to run large scale molecular docking calculations where at a time we'll be docking thousands of molecules using different softwares like Autodoc Vina, SwissDoc, PyRx to screen large number of FDA approved small molecules. to see which ones are showing kinase inhibitory effect. And once you have done that, the next step is to follow it up by simple frequency calculations like MMPBSA, Poisson Boltzmann surface area calculations. These are already implemented in Bromex, this MMPBSA calculation. And once we screen some more molecules off using this frequency calculation, then we'll run something called other frequency MD simulations like metadynamic simulation or umbrella sampling to even further screen more molecules off to study predicate drug interaction. So this is roughly the idea that first we get the enzyme, then we'll screen different library of compounds to identify good number of or good candidate for inhibitors for this particular enzyme. So here we'll be using Bromex MD simulation, molecular docking, and a little bit of free energy calculation because as Sumit sir also said that enhanced sampling calculations are not very easy to run because they require huge number of resources, not only large number of RAM or GPU, they actually require large number of CPUs also, like a large number of nodes where you can actually run many simulations together. Okay, running one single simulation is not always a good idea in these cases. So you need to run like 30, 40 per simulations parallelly. So for that, you need those large computational resources where you can run these things. So we'll try to run single node simulations mostly for this particular project. And in case we have access to those kind of computational resources, we can run one or two in sampling calculators. So with this, I'd like to conclude my presentation. So I would be happy to answer any questions you guys have. Thank you.
Thank you so much. Both of you explained the project very nicely and in very detail. So, Prabhuda, if you have any doubt, you guys can discuss, or you maybe you if you need maybe time to decide yourself which project you want to enroll. You can do it. Maybe it can take 1 or 2 days. So we are planning to start the project from next week onwards, from 15th April onwards. Uh, yes. Okay. Biopsa, you have something to ask?
Yeah.
Yeah.
What I was thinking that this recording will it be shared with other participants as well?
Definitely. Yes. Yeah.
Okay. Yeah. Okay. Great.
Yeah.
Yeah.
No, I mean, even last meeting we did the same way. So I think they are waiting for the recording instead of coming to the live.
Okay. Yeah.
Yeah.
That's
So
nice. Yeah.
yeah,
I think that will be great actually. Yeah. The recordings are available. They can see the slides and
definitely,
everything.
definitely. Yeah, we used to do that. So even the last meeting we shared, and many people watched. Instead of coming to the live, they prefer to watch that recordings. Again, then as it is a recorded word. So again, another instruction you all have to understand. The process of enrolling to a project is first come first basis. So and each project we have very restricted number of students. We cannot accommodate all the people. So you have to understand that point. And some of the projects already complete. The intake is already completed. We already closed some of the projects that you can see in the registration link. Yeah. So now, Prabhuda, if you have any questions, you can ask.
Uh, the thing is, atomistically, we can use CHARM GUI, but as I said, for the membrane model, most likely we are going to use sports grain MD simulation. So there we'll be using uh, Martini force field. But yeah, definitely these membranes can be built up using CHARM GUI. Actually, there are multiple options to do that. You can use something in Martini. There is an option, something called Insane Builder. So using that, you can specify the peptide groups, the lipids that you would like to use, and you can specify their composition or the number. Martini will organize the lipids itself, and it will give you a three-dimensional structure of the lipid bilayer. And also, I think it is possible to prepare these lipid bilayer structures in coarse grain in fashion in Charm GUI also. We'll see to that actually what to use. So we can use both and we can see which one will be easier for us to use. That is the one we'll use. But the idea is to learn not only to do the project, but also to learn how to use these different technologies or different methodologies. Both Charm GUI as well as Charm GUI has more been, I mean, people have more been using it like a black box. I mean, which is fine, not a problem. It's a very good one. But other than that, one should also learn how to use the Martini-based tools like Incense, Script, and all to prepare the membrane structure and everything. So to answer your question, yeah, definitely for some purpose, definitely we'll have to use Charm GUI. But more often than not, for this antimicrobial peptide project, we'll be mostly focused on Martini-based forceful. Yeah, yeah, no problem.
Um, Neither. Can you hear me? So actually, the presentations are over. If you, I don't know whether you went through the abstracts that we already shared, and if you have any questions in those projects, you can ask. Or even if you want watch the presentation, we will definitely upload. I will definitely give this recording, so you can watch it.
The approach of the peptide can also be, one second, can also be applicable for penicillin derivatives. I will have to say to that. I mean, I don't exactly know whether they'll be applicable to penicillin derivatives or not, but I'll have to search literature for that as well. But yeah, I don't have the exact answer, but definitely, I mean, I can give you a lot of examples where people have found, for example, antimicrobial peptides working against E. coli bacteria and other gastrointestinal problem causing bacteria, a lot of different bacteria, for example, antifungal peptides are also possible. Recently, I've been working with a group based in US. They have been developing a medications for dental medications actually. Dental medications, which is an antifungal medications they have been working. That's also antifungal peptide they have been developing. So we are studying the MD simulations of model membranes using their peptide actually. So I think it should work, but I'll have to see. I don't have a conclusive answer on that. Yeah, I understand. I know penicillin is a class of drugs that inhibit the bacterial cell, but what bacterial cell wall synthesis, but I don't know whether this approach, I mean, the mechanism of peptide interaction with the bacterial cell wall and penicillin class of drugs are the same or not. Because the understanding is penicillin is an antibiotics. Okay, is a class of antibiotics, right? So the idea is to study a molecule which is not an antibiotic. I mean, antibiotic. I mean, it will have an antibiotic property, but which is not typical inorganic or organic typical organic small molecule. Okay, we are not going to study that thing. So that is the reason we people have been more focused on using things like antimicrobial peptides, not the traditional antibiotics. Okay, so that is the reason. I mean, penicillin, the mechanisms will be different. So, as if you undertake the project, we'll definitely go through some of the papers. You'll see that antimicrobial peptides, what they do actually, so they disintegrate the membrane in different ways. Some peptide can form a transmembrane pore. So you have the membrane which is intact, but it will form a cylindrical pore inside that membrane. So it will disintegrate that membrane. Some will disintegrate the membrane in a carpet-like fashion. Some will directly interact with the get absorbed on the membrane and completely disrupt the membrane order and everything. So there are multiple toroidal pore formation and there are multiple mechanisms. So that is how it is important to study these things with the specific membrane and specific antimicrobial peptide because the mechanisms can vary. Okay, I hope I could answer some part of your question. Yeah, you're welcome.
Um, yeah, thank you so much, Vivek sir, for answering all the questions. I think Prabhuda has some problem with his microphone. So if we don't have any other questions, then we can wind up the session. Thank you so much, Sumit sir and Vivek sir for explaining the projects. Again, I'm repeating, the number of intake to each project will be restricted. That will follow first come first serve basis. Then yeah, that's it. We thank you all.
Thank
Yeah,
you. Yeah. Thank you,
Thank you
sir.
so
good.
much, sir.
Yeah, good night. Good night. See you.